All right, well, people are still signing on, but I think we'll go ahead and get started. Good afternoon, everyone. This is Chris Newgard at Duke University, and you are signing in to another session of our Pathways Works in Progress series. And we're delighted today to have Mayland, Chang and Maxence Nachury as our presenters. I want to remind everybody that we're going to use the chat system to ask questions. I'll serve as the moderator, and we'll try to get as many of the questions out in front of our speaker and addressed in the limited time that we have. I do hope our speakers have planned on about 20 minutes of presentation to give us time for asking questions. So with no further ado, I'm very pleased to welcome Dr. Mayland Chang today from Notre Dame. She earned an Accelerator Award in early 2015. And she's going to be representing research on diabetic complications in a talk entitled Targeting Matrix Metalloproteinase 9 for the Treatment of Infected Diabetic Foot Ulcers. Mayland, please take it away.
Good afternoon. So today I will talk about my ADA research for the past 5 years, and that dealt with MMP9 for the treatment of diabetic foot ulcers. So I'd like to acknowledge the people in my lab who have done this research: Tran Nguyen, Chi-Hong Peng, Ming Gao, Major Guit, Dirang Ding, Rocio Perez, Bal Schroeder, and Bill Walter. I'd also like to thank my collaborators, Shara Mobashiri at Notre Dame, as well as Charles Peterson and Luis Pantalena at Memorial Hospital here in South Bend for the collection of human tissue used in this work. I'd like to thank the ADA for the generous support, without which this research would not have been possible, as well as my ADA mentor, Larry Chang. I like to disclose that I am a founding member of SalvaPets. This is a startup company that was created to commercialize the development of our MMP9 inhibitor. So diabetes affects almost 400 million people in the world, and that includes 30 million here in the United States. About 25% of diabetic patients develop chronic wounds, typically on the foot, and these are the diabetic foot ulcers. It affects 1 million Americans every year. There's only one approved drug by the FDA called Regranex. Its generic name is becaplirmin, and it contains platelet-derived growth factor, which is an angiogenesis stimulant. It has modest efficacy, so it's seldom used, and there's also increased risk of cancer and death. So the basis for why diabetic foot ulcers are recalcitrant to healing was not well understood. As a result, there are over 100,000 lower limb amputations every year in the United States. And prognosis after amputation is poor, with about 40% of the amputees dying within a year. So wound healing involves an orchestrated set of events that includes inflammation, in which reactive oxygen species and MMPs are upregulated. That is followed by angiogenesis to increase blood flow to the wound, and then remodeling of the extracellular matrix that results in a healed wound. So the problem with chronic wounds, including diabetic foot ulcers, is that they are stalled here in inflammation, and as a result, they do not heal. So if you can see here, MMPs are involved in wound healing. So MMPs are a family of 24 zinc-dependent proteinases whose physiological role is to degrade the extracellular matrix. They are expressed as inactive zymogens called pro-MMPs, in which the prodomain seen here in red covers the catalytic site in yellow. Activation requires removal of this prodomain, to expose the catalytic site, and you can see zinc here in the catalytic site. Now, control of MMPs is highly regulated, and mainly by these protein inhibitors called TIMs, seen here in purple, that cover the catalytic site. So for each of the 24 MMPs, each exists in 3 forms. Only one is active, and the pro-MMPs and the TIM complex MMPs are inactive. The challenge is that most methods do not distinguish between the forms of MMPs, especially between the active and the inactive TIM complex MMP. So to address this, we use this affinity resin that consists of a broad spectrum MMP inhibitor seen here in magenta. It is covalently coupled to Sephiro's resin in blue. This affinity resin binds only to the active forms of MMPs. And the reason for that is that only the active form have the catalytic site exposed to bind to that resin. The pro-MMP have the prodomain covering the catalytic site, and the TIM complex MMP has the TIM covering that catalytic site. So what we do is incubate tissues with the affinity resin. Only the active MMP seen here in yellow bind to that resin. We then reduce cysteines in the MMPs with dithiothioethyl. We alkylate with iodoacetylene, then do a trypsin digestion, and the peptides are analyzed by liquid chromatography, mass spectrometry. Using a protein database, we identify the active MMPs, and then we set up methods to quantify those active MMPs. Okay, so we use the diabetic mouse model, and we create full thickness excision wounds. We infected them with biofilm producing Staphylococcus epidermidis. And when we do that, we see that infected diabetic wounds are delayed wound healing compared to the uninfected ones. We analyze for levels of interleukin-6 as a marker of inflammation, and we see that infected wounds have increased inflammation. We also analyze for levels of vascular endothelial growth factor, and we see that infected wounds, and this is a marker for angiogenesis, infected wounds have low levels of VEGF. We then analyze the wounds with an affinity resin followed by proteomics. We identify only 2 MMPs in their active forms, MMP8 and MMP9. There was no difference in MMP8 between the uninfected and the infected mice, and I'm not showing that data. However, for active MMP9, there was an increase in the infected wounds compared to the uninfected ones. So we collected wound tissue from 25 patients with diabetic foot ulcers, and also from 23 volunteers and healthy controls. We analyzed with our affinity resin, and we identified only 2 MMPs in their active forms, MMP8 and MMP9. So these are the same proteins we had found in diabetic mouse wounds. It indicated that the mechanism of pathology and repair were similar between diabetic humans and diabetic mice. So we classified the wounds by Wagner grade. So a Wagner grade 1 is a superficial ulcer. And as the Wagner grade increases, you have increased severity and infection. So Wagner grade 4 is partial gangrene of the foot, and a Wagner grade 5 is complete gangrene of the foot. We did not have any patients with Wagner grade 5 because these patients have already undergone amputation. So for active MMP8, we found 10-fold higher levels of active MMP8 compared to the control, but it didn't matter the Wagner grade. However, for MMP9, in the less severe wounds, Wagner's grade 1 and 2, levels were 7-fold compared to the controls, and levels increased more than 30-fold in the Wagner's grade 3 and 4. These are the more severe and infected ulcers. And overall, for all the combined Wagner's grade 1 and 4, levels were 20-fold. And notice the Y scale is on the log basis. So we had identified active MMP 8 and 9. The question was, what was the role of each one of these proteins? And to look at the role of MMP8, we use this MMP8 inhibitor that we synthesized in my laboratory. It inhibits MMP8 with a Ki of 25 nanomolar. It inhibits MMP9 poorly with a Ki of 76 micromolar. So when we apply this inhibitor topically to the wounds, the wound closure for the MMP8 inhibitor is in gray and the vehicle is in black. And we see that in the representative photos of the wounds, that when we inhibit MMP8, the wounds get worse, implying that MMP8 has a beneficial role. We confirmed the role of MMP8 by exogenous application of recombinant mouse MMP8. The gene was cloned in my lab, and we expressed and purified the protein. And when we applied topically excess MMP8, the wound healing was accelerated. This indicated that MMP8 plays a beneficial role in wound healing. So to look at the role of MMP9, we use this inhibitor called ND322 that was discovered in my laboratory. It inhibits MMP9 with a Ki of 870 nanomolar and inhibits MMP8 with a Ki of 2.6 micromolar. So when we applied ND322 topically, the wounds got better, as seen by these pictures, and the wound closure here, where ND322 is in the open circles. To confirm the detrimental role of MMP9, we use MMP9 knockout mice that we made diabetic by treatment with streptozotocin. And when we ablate MMP9, we found that wound healing was accelerated, indicating that MMP9 played a detrimental role. So our research had shown that MMP8 is beneficial and MMP9 is detrimental to wound healing. So the best strategy would be to inhibit the detrimental MMP9 without affecting the beneficial MMP8. So then we started a lead optimization program. And these are the structures of over the 450 compounds that we synthesized. We discovered a selective MMP9 inhibitor that we call RND336. It inhibits MMP9 with a Ki of 19 nanomolar. It's a mechanism-based low binding inhibitor. So at the active site of MMP9, this reaction takes place in which the thyrane, so this is the sulfur containing ring opens to the corresponding thiolate, and this binds zinc. The reversal occurs very slowly. As a result, the residence time, this is the time RND336 is bound to MMP9, is very long, in the order of 5 hours. In contrast, it inhibits MMP8 poorly with a Ki of 8,600 nanomolar, and the mechanism of inhibition is different as a linear non-competitive inhibitor, for which the residence time is in the order of seconds. Now, in the environment of the diabetic wound, RND336 only inhibits MMP9. It is water soluble, about 5 mg per ml. And if we compare to the natural TIMP inhibitors, time is about 7 to 8 minutes. So the synthetic inhibitor is much better than the natural TIMP inhibitor. So we then evaluated RND336 side by side with becaplirmin. Becaplirmin is the only FDA-approved drug. So the wound closure here in blue is vehicle, in red is RND336, and becaplirmin is in black. And we can see that wound healing is accelerated in RND336 compared to becaplirmin, as seen by this representative figure, photos of the wounds. We look at inhibition of MMP9 activity by in situ cymography using a fluorescence DQ gelatin for MMP9 activity. So MMP9 activity is seen as green. In blue is nuclear staining by DAPI. So when we apply becaplirmin to the wounds, we see that becaplirmin decreases MMP9 activity, but it does not completely inhibit it like RND336. And this is how we explain the better efficacy of RND336. We analyzed the wounds with our affinity resin followed by proteomics. There was no difference in MMP9. I'm not showing that data. However, for active MMP9, we see that becaplirmin reduced the levels of active MMP9. So most human wounds are infected. We thus evaluated the efficacy of RND336 in the infected diabetic mouse wound model. And we have 4 treatment groups. In black is vehicle. In blue is the antibiotic linezolid. In red is RND336. And in green is the combine of RND336 and linezolid. So linezolid by itself has no efficacy compared to vehicle control. However, wounds treated with RND336 in red or the combination showed acceleration of wound healing as seen in these representative photographs. And the combined treatment appeared to be better than either single treatment alone. So the wounds treated with RND336 and the combination show complete reepithelialization compared to partial reepithelialization, and this is shown by the white line for vehicle and linezolid. We look at the bacterial load in the wounds, and we see that only the wounds treated with linezolid or the combination of RND336 and linezolid had 2 log reduction in colony-forming units. We also analyzed for BGF as a marker for angiogenesis, and we see that the wounds treated with either RND336 or the combination showed increased angiogenesis. We look at in situ cymography for MMP9 activity, where MMP9 activity is seen in green. The blue is nuclear staining with DAPI. And we also stain for macrophages with F480, and this is shown in red. So if we look on top here, the vehicle, we see co-localization of MMP9 activity with macrophages. When we treat with RND336 or the combination, we see complete inhibition of MMP9 activity. And also we see reduction in macrophage infiltration. So that RND336 is inhibiting the detrimental MMP9. It mitigates macrophage infiltration. It also increases angiogenesis, therefore restoring the normal wound healing process. So in summary, upon injury, neutrophils come to the wound. They secrete MMP8, and that is depicted with this blue color. And MMP8 repairs collagen, remodels the extracellular matrix, and heals the wound. Neutrophils also secrete ROS that activates NF-kappa beta, and that activates MMP9. So excess MMP9 destroys the extracellular matrix and keeps the wound from healing. So we also see that monocytes, they get converted to macrophages and they release TGF and BGF that improve angiogenesis. So we think the best strategy is to selectively inhibit the detrimental MMP9 with the small molecule RND336 so that remodeling of the extracellular matrix can take place to heal the wound. And we have seen in humans that the more severe and infected wounds have increased levels of active MMP9. So where are we now? We're currently conducting investigational new drug enabling studies for RND336 for treating diabetic foot ulcers. These include the GLP toxicology, the chemistry, the control, manufacturing, and the chemistry for the drug, as well as GMP manufacturing. This has been done with help from a Therapeutic Development Award from the Department of Defense. And we are anticipating starting phase 1 clinical trials next year. This concludes my presentation. I'd like to thank you for your attention, and I'll be happy to answer any questions.
Thank you, Mayland. That was terrific. Please, everyone on here, send in any questions by chat. But let me go ahead and start. So let me be clear. Becaplamine, sorry, did I say that right?
Yes.
Becker, yeah. Is that a clinically approved product at this point? That's an alternate MMP9 inhibitor. Is that not correct?
It is approved by the FDA. It was approved more than 20 years ago. Now, we found that the way it's working is through indirectly decreasing MMP9. So it's a MMP9, it's a poor MMP9 inhibitor. So when we look at kinetics of becaplermin, we get very poor inhibition. I think it's about 17%, but at 4 micromolar, okay? So in here, we're getting Ki for RND336 is 19 nanomolar. So it's a much better inhibitor.
Right.
But it is, we think that becaplermin is acting way. But because it is not directly inhibiting MMP9, in clinical trials, it takes about 10 weeks for becaplermin to start showing differentiating from placebo.
Okay. Rohit Kulkarni asks, do any of these inhibitors also restore sensation? In other words, nerve loss in the wounded area?
Yeah.
And are MMPs involved in any nerve actions?
We have not looked at that. Now, MMP9 is implicated in many neurological diseases, so it would not be surprising that it may be involved in that, but we have not looked at that.
Okay, and I was wondering, I think all the models that you showed were diabetic hyperglycemic models where you're testing all your agents. What is the role of hyperglycemia in terms of the infection severity, and do some of the compounds that you've been developed, or does it require a background of hyperglycemia to show the kind of wound healing efficacy that you demonstrated today? In other words, if you created a wound and infected it as a researcher, do the MMP9 inhibitors work just as well in a non-diabetic as they do in a diabetic setting?
No. So if we've done actually this in non-diabetic mice, when we make the wounds, there is really no MMP9. So when you apply an MMP9 inhibitor, it does nothing because MMP9 is not upregulated.
I see. I
Okay, so you must have a diabetic mouse for that to work. Mm-hmm.
Okay, here's a question from Chris Rhodes. Does netosis, in parentheses, or in air quotes, play a role in diabetic wound healing? If so, are MMPs involved? I hope you understand what netosis is.
No, I don't.
Oh, good. Chris, can you come on and tell us what netosis is? Can you unmute?
Yes, I am. Can you hear me?
Yeah, go ahead.
Yeah, I mean, a really interesting piece of research that you're doing here and really fulfilling an unmet need. But NETosis is sort of like a web generated by neutrophils that catches the bacterial infection in place. And, and and some people think it might make it better because it attracts macrophages to deal with it, and some people might think it might worse by keeping the bacteria there and just enhancing the infection and prolonging it.
Yeah. So
So
we have looked at the.
Perhaps you shouldn't answer it.
We've looked at the effect of RND336 of our small molecule inhibitors, and they're not. So we look at MIC, the minimum inhibitory concentration. They're not antibacterial. But in a number of diseases where there is infection, there is MMP9 that is upregulated. So not just infected diabetic wounds, but other in general infections increase MMP9 levels. So for example, it has just been reported that in COVID infected patients, they see increase of MMP9. So now I'm not sure if that is active because they usually do by ELISA, and ELISA captures both 3 forms of MMPs. But that would be an interesting hypothesis to look at.
Okay, and one more question from Ernesto Bernal Mizrachi. Do these inhibitors enhance angiogenesis?
They do temporarily. So we always look at as a as a function of during the course of the wound healing. So we only get temporary increase of angiogenesis and decrease of inflammation, okay, to restore the wound healing process. Now, if you continuously have increased angiogenesis, we don't think that is good. That is what becapleurin does. And that will not be good, especially in the more senior population of the diabetic patients, in which maybe they're more propense to get cancer. So that would not be a good thing. So these inhibitors just temporarily improve angiogenesis just to restore the wound to the normal healing process.
All right. Well, Mayland, not only was that clear and translationally exciting, but you kept to time. We are very grateful. Thank you so much.
Thank you.
Beautiful job. And we'll move on then to our next presentation, which will be by Maxence Nachury from University of California, San Francisco. He's very new to the fold. He came in as a Visionary Award winner at the beginning of 2020. And he's going to give us a talk about regulation of energy metabolism by signaling and trafficking at the primary cilium. Maxence, please.
All right. Thank you, Chris. Thank you all for joining me. Mayland plays the bar very high there, and I'll do my best to keep on time as well as she did now. So I'm going to tell you today about a project that really started when I moved my lab from Stanford to UCSF about 3 years ago. And that started really as a collaboration with the lab of Christian Vest here in the Diabetes Center. My background is really as a mechanistic cell biologist. So I'm interested in figuring out how things work inside the cell. And it's taken me all the way now to studying the regulation of energy homeostasis, and something I'm not going to tell you about today, but also beta cell function. So about 15 years ago, I became interested in this little part of the cell called the cilium. So the cilium, you might know about motile cilia that line your airways, a beat. That's really important if you want to keep pathogens outside of your lungs. But it turns out that there is a counterpart to these motile cilia that is present on nearly every cell in your body, and that's called the primary cilium. So this primary cilium in particular, it's present in every neuron in your brain, for example. It's present in your alpha cell, beta cell, delta cells in your pancreas. And it's in fact a lot more interesting than motile cilia because they do signaling. And nowhere is it better illustrated than in the sense of vision, where every photon that hits your retina hits a G protein coupled receptor, rhodopsin, that's concentrated right here in a super specialized primary cilium. And together with this GPCR, you have the entire signal transduction cascade all the way down to the cyclic nucleotide gated channel. So what comes out of the cilium is an electrical signal in this case. Olfaction works following a very similar paradigm. But the role of the cilium in signaling isn't limited to sensory signaling. It's also true in the case of developmental signaling. And there it's best illustrated by hedgehog signaling, which is a pathway that patterns your neural tube. And here you can see again this paradigm of concentration of a signaling cascade in the primary cilium with one twist, which is that the concentration there is dynamic. The concentration of these various GPCR signaling receptors is gated by pathway activation. And it's really important because the dynamic trafficking of these signaling molecules is integral to the transduction of hedgehog signal, in that in this state, the cilium bears a high concentration of cyclic AMP, and there a low concentration of cyclic AMP. That will commit a transcription factor for contrasting fates, in this case, a repressor processing, in this case, an activator processing. And there you have the basis for the whole transcriptional program engagement downstream of hedgehog signal. But this is really the tip of the iceberg here that I'm showing you, because if you survey the non-olfactory, non-photosensitive GPCRs, you can see that you have about 10% of them that reside in cilia. And if that wasn't enough to convince you that cilia are important in physiology, the ciliopathies should convince you, because here is a host of about 20 different disorders that are characterized by mutation in over 100 different genes, where you get patients that have retinal degeneration, kidney abnormalities, developmental delay, skeletal malformation, and what I'm most interested in right now is obesity. And the one ciliopathy that we've studied for the past 10 plus years is called Bardet-Biedl syndrome. Let me see here. Here we go. So Bardet-Biedl syndrome encompasses most of the symptoms that I just mentioned. One thing I should say about obesity is that studies in mice have really cemented the hypothesis that ciliary dysfunction underlies a wide range of cases of obesity. So if you ablate cilia in the adult brain, mice become obese. You can also ablate them in specific region of the brain, and again, you get obesity. In human genetics, one thing that you might be familiar with is GWAS studies that have identified a polymorphism at the FTO locus. Well, studies by Rudy Leibel at Columbia have shown that this polymorphism is likely to decrease the expression of a gene that's required for the structure of the cilium. And so the hypothesis here is that in this part of the population that has the polymorphism as the FTO locus, their cilium function would be negatively affected. So Bardet-Biedl syndrome, I'm going to sum up work that we've done over the past 15 years. And this is really just to say that what the basis of Bardet-Biedl syndrome is within this complex of 8 Bardet-Biedl syndrome proteins that's called the BBsome, functions together with another BBS gene, which is R6. And together, what those things do is that they take activated GPCRs outside of cilia by allowing them to pass through this diffusion barrier here at the base of the cilia. One of the questions that was asked repeatedly when I would present this data is, well, how can it be that you get a full organism that develops when you cannot remove things from cilia? How are ciliary exit defects tolerated? And that's one question that Drew Nager in the lab got interested in. And what he found was that when the BBsome function is disrupted, here we're taking this GPCR, somatostatin receptor 3, it doesn't exit cilia, but appears to accumulate in these little bulbs at the tip. And there Drew, in a stroke of genius, decided to image in real time what's happened. And what he found is that these bulbs detach from the cilium in a process that is named ectocytosis. So it shouldn't be confused with exocytosis. Ectocytosis really describes this movement of vesicles outside of the cell. So that's the explanation for why organisms can tolerate loss of BBsome, is that they have a safety valve being ectocytosis. Ectocytosis is a very efficient process. So with each round of ectocytosis, you can read the cilium of as much as 1/3 of its pool of active GPCR. But there is a cost. With each ectocytosis event, the cilium consumes material, so you end up getting cilia that become, over time, shorter and shorter. So a much less ordered process than the BBsome-mediated retrieval. So how does it work? What are the mechanisms of ectocytosis? At this stage, we don't know very much. The little bit that we know came from studies using cilia proteomics that David Mick in the lab did a few years back. And there, this really is something that was born out of the limitations that we cannot purify cilia. So we had to find another way. And the other way is that we target this enzyme, a peroxidase to cilia, and now we can biotinylate the entire content of the cilium, which allows us to define the proteome of the cilia. Now we can use this technique to profile the changes of the proteome. And in particular, we looked in cells where ectocytosis is overactive. This is a specific BBS mutant here. And what we found is things that we already knew accumulated in cilia of this mutant, which is BBsome and BBsome cargos. But surprisingly, we found actin regulators, particular myosin 6 and drebrin. And here, Drew was able to see that myosin 6 itself is present as the prospective site of ectocytosis right at the tip of the cilia. And functionally, when he either pharmacologically interfered or genetically interfered with these actin regulators, he could see that ectocytosis was strongly inhibited, but that retrieval function was normal. So that's my introduction for you, is that there are really two means that the cilium has to get rid of unwanted proteins. One of them is this ordered pathway of BBsome-mediated retrieval, and the other one is this pathway of ectocytosis. So the question that we wanted to answer is, how does ciliary trafficking influence body weight homeostasis? What goes wrong in BBS patients that causes this uncontrolled weight gain? And in order to answer this question, what we need to know is, what is the ciliary pathway that limits obesity? So body weight homeostasis is something that's absolutely remarkable, in that we ingest roughly a metric ton of food a year. And if things go okay with this pathway here, we don't gain much weight. So there is an incredibly well-tuned rheostat here that will limit food intake depending on the amount of energy that is stored in your body. And in particular, this hormone leptin that's secreted by fat store is sensed within the brain in regions of the hypothalamus called the arcuate nucleus that will then influence the status of the second-order neurons in the paraventricular nucleus to regulate satiety. You're going to feel, if you store a lot of fat, you're going to feel full earlier. That's what this system does. And so over time, people have looked at the various components of this regulatory pathway and have found one GPCR that is localized to cilia, and that's a neuropeptide-wide receptor 2. So NPY2R, you can see Peter Jackson's lab published a very nice paper, and we confirmed those findings. So NPY2R does localize to cilia. The problem, though, is that the knockouts of NPY2R don't exhibit much of a phenotype. They don't have obesity. So what else is there? Well, what else is there is the melanocortin-4 receptor. And here, if you think in terms of human genetics about the greatest contributor to obesity, that's MC4R. In fact, if you lose just one copy of MC4R, you're already going to be morbidly obese. And you can see in the morbidly obese population that you have about 5% of patients that harbor mutations in MC4. So MC4 is a little unusual in the realm of G protein coupled receptor, is that it's regulated both by an agonist, alpha MSH, as well as a negative regulator, AGRP, the agouti-related peptide. But the bottom line is that when MC4 is active, satiety ensues. And what Christian Vest described a few years back is that MC4 is localized to cilia in the neuron of the paraventricular nucleus. So this is where we came in and we decided to study the regulated trafficking of MC4. So the first thing that we did, and all the work I'm going to present today was done by Irene Ojeda-Najaos, was first to reconstitute MC4 trafficking in cell culture. And you can see here, you have beautiful localization of MC4 to cilia. One question, though, is that other people had looked for MC4 in cilia and hadn't seen it. So what was the difference? Well, the difference here really was, again, thanks to Christian's lab, the identification of this protein called MRAP2, MC4R associated protein 2. Well, that's a single pass transmembrane protein. And you can see that if you don't express MRAP2 together with MC4, there isn't much MC4 in cilia. You co-express those two things, and they both beautifully go to cilia. So this is the answer, is that MC4 appears to function as a heterodimer together with MRAP2. And if you have mutation in MRAP2, same as with MC4, you get a dramatic weight gain. Okay, so now we had reconstituted MC4 localization to cilia. Does it undergo dynamic trafficking? And the answer is yes. You can see here. And I should just mention, you're going to see that on a few slides, is that in the main image, you're going to have the channel overlapping. And there we just shift the channel so that you can better distinguish the two channels. So MC4 here in yellow, you can see it there. You treat with agonist, it's gone. But there was one thing that was surprising that Irene found, is that first of all, the cilia start out fairly short and they get even shorter when she stimulates. And that's not something that we normally see. So if you take this run-of-the-mill GPCR there that exits cilia through the BBSO-mediated retrieval pathway, the length stays constant. So by now, you're all professionals of ectocytosis, and you know that this shortening of cilia is a telltale sign of ectocytosis. So this led to the hypothesis that MC4R, as its default pathway, was using ectocytosis. And that was really surprising because so far we had only viewed ectocytosis as a fail-safe for exit from cilia. So we decided to really drill down into this and determine if really MC4 is exiting cilia via ectocytosis. So the first thing is that we know these two pathways, ectocytosis, retrieval. If really MC4 exits via ectocytosis, it shouldn't care about the retrieval pathway. And indeed, that's the case. You can see MC4 exits normally here. It still exits normally when BBsome function is abrogated. Second thing is that we know that actin is required for ectocytosis. Now, it's a fairly blunt instrument to treat cells with actin depolymerizing agent, but at least it doesn't interfere with retrieval from cilia. And what we saw here is that it does interfere with MC4 exit from cilia. You can see MC4 here. You treat cells with this actin poison, it cannot exit cilia. So that was the second piece of evidence. The third thing we can do is that we can purify extracellular vesicles. And these are established protocols. You just take your tissue culture supernatant, subject it to several steps of differential centrifugation, and you can harvest extracellular vesicles. So this is from past studies here, but you can see these extracellular vesicles, they're really packaged with GPCRs, and they have sizes that are just under 100 nanometers. And when Irene did the experiment here with cells expressing MC4 and stimulated with the agonist, she saw that there was a dramatic increase of MC4 in extracellular vesicle, roughly between 50 and 100-fold increase upon MC4 activation. And the final piece of evidence is that we can directly image MC4 and see that it is getting released in extracellular vesicles. All right, so that's really the heart of my presentation today, which is that MC4 leaves cilia, activated MC4 leaves cilia via ectocytosis. So in the past, we've always seen ectocytosis as a mean to dispose unwanted proteins. But the fact that a protein, that a GPCR can utilize this pathway as its default exit pathway suggests that it may be doing more than being a mean of disposal. So a major goal now, and that's really work in progress, is to address the function of these MC4 EVs. And so we're collecting various data sets from proteomics, either directly of EVs, of cilia that are undergoing ectocytosis, to hopefully get insights into the molecular mechanism of ectocytosis so that we can block ectocytosis in these MC4 neurons and assess feeding behavior and body weight homeostasis. So one thing I'm going to show you, again, as work in progress that we're trying in this direction, is leveraging this knowledge that actin polymerization is important for ectocytosis. And here what we're doing is that we're taking a protein that severs actin filament called gelsolin, and we're targeting this protein only to cilia. So that's what we call the genetically encoded modifiers of ectocytosis. And this gene, when we transfect it into cells, you can see that here this ectocytosis cargo, because the cells are BBS null, now is no longer able to exit cilia. So it's not perfect. We need to express this at very high levels. So we're not quite satisfied with it yet, but these are the ideas here that we're having, that we can take activities that might be very detrimental to the cell, but if we target them very specifically to cilia, we hope to be able to specifically interfere with this process of ectocytosis. All right, so this is pretty much what I had for today. Just wrapping up, the BBsome, I've told you that it's retrieved activated GPCR. We, until recently, thought that ectocytosis was just a safety valve, but it turned out that MC4 really is change the studies of MC4R is changing our views in that it appears to use ectocytosis as its default exit pathway. The other thing that's interesting is that it is co-targeted and seems to co-function with a single pass membrane proteins. But this discovery that that MC4 undergoes signal-dependent ectocytosis is really something that we hope to be able to leverage in the future to limit body weight gain in obese patients. And that's going to require more knowledge, because at this point, we don't even know yet if these MC4 EVs are bioactive, if they're taken up by other cells, if they're doing something in other cells, or if it's really just a means of disposal. So that's really the first thing we need to figure out before we can consider any type of intervention. But I think the question I posed at the beginning, which is what is the basis of obesity in BBS patients, we still have not answered it. And in fact, this mechanism that we're seeing for MC4 is completely BBsome independent. So that is still a major question. I'll be happy to discuss that if you're interested. But for now, I just wrap up. I thank the people who've done some of the work in the past: Drew Nager, David Mick, David, who now has his own lab in Germany, and Irene, who's been really instrumental to this project. She's now joined by an undergrad, Ralph, another postdoc, Mingli, and again, none of this work would have been possible without Christian's really getting me into this field and the ADA for funding all this work. So thank you.
Fantastic, really interesting, and a flurry of questions came up. So here we go. Rohit again, interesting talk. What's the timeline for an event of ectocytosis? Is it minutes or seconds? And how many receptors can be removed in one event of ectocytosis?
Okay, so great question. Yeah, so it's fairly infrequent. So ectocytosis, unlike retrieval just there is a steady stream of retrievals that takes place once you activate a given GPCR. Ectocytosis, typically within the span of 2 hours, we could detect one event per cillian. So that's about the time frame. Now, in terms of the magnitude, we know that it takes out a third of the GPCR pool from cilia. And GPCR pools, typically, we've done molecular counting, they number in the 1,000. So I would say it could take as many as 300 molecules, but I don't think all these 300 are in one vesicle. It's likely that these events that we identified by microscopy, they might release several vesicles. So, but yeah, roughly one event we could detect would take 300 molecules.
Yeah, thanks very much. The reason I asked is whether these processes are subject to regulation, dynamic regulation, which your overall goal is obesity. So I was just curious to know overall, how does it factor into the regulatory process?
Well, so the one thing we know regulates it is the state of activation. So if we activate MC4R, it goes into ectosomes. If and I didn't show that today, but if we take an antagonist or an inverse agonist, it does not go into ectosomes. So clearly that's gated by the state of activation. Yeah.
Thank you.
Okay, thanks. So Alan Saltil has a question. What % of total MCR4 is found in the cilia?
That's a really good question. I don't think that's one we can really answer very well. We know that functionally it needs to be in cilia in order to function. So what Christian had found is that mutations that are found in patients that were completely unexplained until now, these mutations blocked ciliary trafficking. Everything else was okay. They could traffic to the cell surface in HEK cells. They could activate GS in HEK cells. They could do all the things that people had SA, but they can't get to the cilium. So it appears that it needs to go to the cilium. Now, what fraction goes there? Yeah, that's a tough question to answer. And we could do that because we can do molecular counting of fluorescence, and we could do quantitative Western blot. So I think we could do that, and we probably should do it. But yeah, right now, not something we have an answer.
Okay, from John Campbell, fascinating talk. How do synaptically released neuropeptides like AGRP and alpha MSH reach the MC4R on the primary cilia, or do you think that there may be extrasynaptic release of these peptides?
Yeah, so my understanding is that these are volume synapses, like dopamine synapses, where the neuropeptide can explore more than just an intersynaptic space there. Now, there are studies that are trying to really get at the architecture of cilia in the brain. Everything is so packed that it's really hard to say anything. What I can say is that what no one has seen is a synapse being opposed to the cilium. So that's definitely not the case. So there must be some diffusion out of the synaptic cleft of these neuropeptides.
And I'll ask the last question then, sort of from the dumb physiologist. Are there changes in the dynamics of this system with, old-fashioned physiologic interventions like fasting, or perhaps even starvation over a couple of days? Does the whole system speed up or slow down under those circumstances?
Yeah, so this is work that Christian has been doing there. And yeah, very clearly, if you fast the animal, you can start to see changes in the level of MC4 in cilia. In fact, the one thing that really puzzled Christian for the longest time is that if you look in adult mice, there is extremely little MC4 in cilia. It's almost undetectable. So it appears that the pathway here is constantly stimulated, and that's with mice fed high-fat diet, I should mention. So in this system, when MC4 is constantly stimulated, there is extremely little that's in cilia, probably because it is undergoing constant ectocytosis. And so one idea is that if really it's functioning in cilia of MC4 neuron, if we could block ectocytosis, we might increase the amount of bioavailable MC4R and potentiate the effect of MC4 agonist for the treatment of obesity.
All right, we'll bring it to a close now. Fantastic lectures today for Mayland and Maxence. Thank you very much.
Thank you.
I want to remind everybody that our next session is Wednesday, March 3rd at 2 p.m. and our speakers will be Michael Dennis and Alex Nektow. I really love the engagement of everyone. Thanks to our speakers and to our participants. See you in a month. Take care.
Thank you.